This video is audio content only. It does not contain any video footage or images. If you are expecting visual content, please be aware. This is intentionally provided in an audio-only format. We recommend listening to the audio rather than watching the screen. Thank you for your understanding. Welcome to a fascinating journey into the intricate world of human genetics. We often focus on the obvious, but today we're diving deeper into a less discussed aspect of our biological blueprint. Join us as we uncover the surprising complexities and hidden challenges associated with having two X chromosomes. From subtle genetic imbalances to profound impacts on health, development, and even longevity, prepare to see this fundamental genetic difference in a whole new light. This narration will reveal the intricate biological trade-offs that shape us. Chapter 1: The X Chromosome's Dosage Dilemma. Our bodies are intricate tapestries woven from countless cells, each containing a precise instruction manual for life, our chromosomes. Most human cells are home to 46 of these thread-like structures, neatly organized into 23 pairs. 22 of these pairs, known as autosomes, are virtually identical in everyone, regardless of their biological sex. But it's that final 23rd pair, the sex chromosomes, that introduces a fascinating and often complex distinction. For most of us, this pair comes in one of two common configurations, either two X chromosomes, typically found in females, or one X and one Y chromosome, typically found in males. Crucially, at least one X chromosome is essential for human survival. Individuals without an X chromosome are not viable. This seemingly simple difference, however, sets the stage for a profound biological challenge, particularly for those with two X chromosomes. Imagine the X chromosome as a grand library, vast and filled with hundreds, even thousands of essential instruction books. Now, picture the Y chromosome as a much smaller, specialized annex containing only a handful of manuals, mostly focused on male development. The X chromosome is indeed a genetic powerhouse, spanning approximately 155 million DNA building blocks and housing an estimated 900 to 1,400 genes, each providing instructions for making vital proteins that perform a myriad of roles throughout the body. In stark contrast, the Y chromosome is significantly smaller, about a third of the X's length, and carries a mere 60 to 70 protein-coding genes, with many of these specifically active in sperm-producing organs. This dramatic disparity in genetic content between the X and Y chromosomes presents an immediate biological puzzle. If males have just one X chromosome and females have two, wouldn't females end up producing twice as much of every protein encoded by the X chromosome? In the finely tuned machinery of our cells, where the precise amount of each protein is crucial for optimal function, such a double dose could be highly disruptive, even harmful. This is what scientists refer to as the dosage problem. For many genes, the quantity of their protein product must fall within a very specific range. Too much or too little can throw the entire system out of balance. These are known as dosage-sensitive genes. Think of it like building a complex engine. An extra spark plug might not matter much, but an extra piston could cause catastrophic failure. The existence of numerous dosage-sensitive genes on the X chromosome means that a direct double dose in females would create a significant biological hurdle. Over millions of years of evolution, life has devised ingenious solutions to such fundamental problems. For the X chromosome dosage imbalance, the solution is a remarkable process called X-Inactivation, or lionization, first hypothesized by Mary Lyon in 1961. Early in the embryonic development of a female, when the embryo consists of just a few hundred cells, one of the two X chromosomes in each cell undergoes a profound transformation. This process is initiated by a long non-coding RNA called X-IST, which coats one of the two X chromosomes, leading to its largely silenced state. This effectively shuts down the chromosome to prevent the overexpression of its genes, ensuring that, like males, females generally have only one active X chromosome per cell. The silenced X chromosome doesn't disappear. Instead, it condenses into a compact, dense structure known as a bar body, which can often be observed under a microscope within the cell's nucleus. It's like muting a television. You can still see the picture, but you can't hear the sound. The muted X chromosome is still present, but its genes are mostly quiet. The process of X-Inactivation is not only elegant, but also typically random. In most cells, it's a matter of chance whether the X chromosome inherited from the mother or the X chromosome inherited from the father is the one that gets inactivated. This randomness creates a fascinating biological mosaic within every female. Her body becomes a patchwork of cells, some where the maternal X is active and others where the paternal X takes the lead. This cellular diversity can have far-reaching implications, influencing everything from subtle physical traits to responses to disease. For instance, the distinctive coat patterns of calico and tortoiseshell cats, which are almost exclusively female, are a classic visual example of this random X inactivation at play, with different patches of fur expressing genes from different active X chromosomes. However, this sophisticated dosage compensation system is not entirely foolproof, and herein lies the inherent challenge. X-inactivation is not a complete or perfect silencing of the entire chromosome. A significant portion of genes, estimated to be around 12 to 15%, consistently escape inactivation. This means they remain active on both X chromosomes in females, leading to a higher level of expression compared to males who only have one X. Many of these escapee genes are located in specific regions at the ends of the X chromosome, known as pseudo-autosomal regions. These regions are unique because they share similarities with genes found on the Y chromosome, meaning both males and females typically have two functional copies of these genes. Beyond these specific regions, an additional 8 to 10% of X-linked genes show variable escape, meaning their activity on the inactive X can differ depending on the tissue type or even from one individual to another. The existence of these escape genes means that females, despite X inactivation, do not have a perfectly balanced dosage of X-linked genes compared to males. They still have a higher expression of these particular genes, which can contribute to subtle yet significant differences between the sexes. This imperfect solution to the dosage problem is vividly illustrated by conditions where the number of X chromosomes deviates from the typical 2 in females or 1 in males. These are known as sex chromosome aneuploidies. Crucially, these sex chromosome aneuploidies are generally less severe than aneuploidies involving autosomal chromosomes, like trisomy 21 or Down syndrome, precisely because of the partial dosage compensation offered by X inactivation. Consider Turner syndrome, a condition affecting approximately 1 in 2,500 live female births. Individuals with Turner syndrome typically have only one X chromosome, often written as 45X, rather than the usual 2. If X inactivation were a perfect silencing mechanism, one might wonder why having only one X chromosome would cause any problems, since females normally only use one active X anyway. The answer lies precisely in those genes that escape inactivation. With only one X chromosome, individuals with Turner syndrome have only one copy of these critically important escapee genes, leading to an insufficient dose. This underdosing contributes to a range of characteristic features. On the other end of the spectrum is triple X syndrome, also known as trisomy X, 47XXX, which occurs in about 1 in 1,000 females. Here, individuals have 3 X chromosomes instead of 2. Again, if X inactivation were perfect, one might expect the extra X to be fully silenced, leading to no discernible effects. However, because genes escape inactivation, having 3 X chromosomes means an overdosing of these specific genes, leading to a distinct set of characteristics. Even in males, the presence of an extra X chromosome, as seen in Klinefelter syndrome, 47XXY, which affects approximately 1 in 600 males, underscores the inherent challenge. Here, males have 1 Y and 2 X chromosomes. One of the X chromosomes undergoes inactivation, yet the remaining active X and the incompletely silenced additional X still lead to a higher dosage of escapee genes than in typical XY males. This higher dosage of escapee genes influences development and health, demonstrating that even in the context of X-Inactivation, the precise number of X chromosomes and the genes that remain active on them profoundly influences development and health. So, while X-Inactivation is a powerful biological mechanism that largely balances the genetic load of the X chromosome between the sexes, it is not an absolute equalizer. The persistent activity of certain genes on the inactivated X chromosome means that females operate with a unique genetic landscape, one where gene dosage is subtly yet significantly different from males. This inherent biological design, a mixture of elegant compensation and persistent dosage differences, creates a foundation for both resilience and vulnerability that shapes female health in ways we are only beginning to fully comprehend. The next chapter will delve into the specific genes that defy this inactivation and the consequences they bring. Chapter 2: Escapees. X-Inactivation's Unsung Genes. While an ingenious evolutionary strategy to balance genetic output is not a perfect silencing mechanism, the persistent activity of genes that escape inactivation introduces a layer of complexity that profoundly impacts female biology and health. This means that females, despite X-Inactivation, don't truly operate with a single dose of X-linked genes across the board. Instead, they carry a double dose of these escapee genes, leading to a higher level of expression compared to males, who only possess 1 X chromosome. Many of these persistent genes are found in special regions at the very ends of the X chromosome, known as pseudo-autosomal regions. Genes within these regions, particularly in what scientists call PAR1, are crucial for normal development. Beyond the pseudo-autosomal regions, other X-linked genes that have active counterparts on the Y chromosome also often escape inactivation. These XY homolog pairs, such as DDX3X, KDM6A, and ZFX, also contribute to the higher gene dosage seen in females compared to males for these specific genes. The impact of these escapee genes is perhaps most strikingly visible in individuals with sex chromosome aneuploidies. These conditions offer a clear window into the consequences of X-Inactivation's imperfection. Take, for instance, Turner syndrome, where a female has only 1 X chromosome instead of 2. Because they only have 1 X chromosome, individuals with Turner syndrome have only a single copy of these normally double-dosed escapee genes. This underdosing of critical genes leads to a deficiency that manifests as specific health challenges. A prime example is the SHOX gene, located in a pseudo-autosomal region and found on both the X and the Y chromosome. This gene plays a vital role in skeletal development, particularly in the growth of arms and legs. In typical females, 2 copies of SHOX are active. In Turner syndrome, only 1 copy is present and active, leading to an insufficient amount of its protein product, which is a major contributor to the distinctive reduced height observed in affected individuals, who average around 4 feet 7 inches tall. Furthermore, the XIS gene, crucial for X-Inactivation, is downregulated in Turner syndrome, contributing to the overall genetic imbalance. Conversely, the TSIX gene, which plays an opposite role to XIS, is consequently upregulated. Conversely, consider conditions where there are extra X chromosomes. In triple X syndrome, a female has 3 X chromosomes, XXX. While one X chromosome is inactivated, the remaining 2 active X chromosomes still lead to a triple dose of those genes that escape inactivation. Similarly, in Klinefelter syndrome, males have an extra X chromosome, XXY. Here, too, one X is inactivated, but the remaining 2 active X's contribute to an increased dosage of escapee genes. This overdosing of certain escapee genes can lead to a range of developmental and neurocognitive challenges, which are explored in more detail in Chapter 4. In these conditions, the XIST gene is often upregulated, reflecting the body's attempt to inactivate the extra X chromosomes. Correspondingly, the TSIX gene, which normally counteracts XIST, is downregulated. The impact of extra X chromosomes becomes even more pronounced with greater numbers. Consider conditions like 48 XXXY syndrome or 49 XXXXY syndrome. In these rare cases, individuals have 3 or even 4 extra X chromosomes, respectively, alongside a single Y chromosome. While all but one X chromosome are inactivated, the cumulative effect of multiple active X chromosomes, each contributing its share of escapee genes, leads to more severe intellectual disabilities, developmental delays, and physical differences compared to Klinefelter syndrome. The extra genetic material from the pseudo-autosomal regions, as well as other XY homolog pairs, likely contributes to the more profound symptoms. The intricate balance of gene expression, so vital for development, is significantly disrupted by these additional copies, even when the body attempts to compensate through inactivation. Beyond these broad impacts, research is pinpointing specific escapee genes and their roles in these conditions. For instance, the KDM6A gene, which escapes inactivation, is involved in modifying DNA and plays a role in the development of gametes, contributing to the reduced fertility often seen in both Turner and Klinefelter syndromes. Another escapee, the SLC16A2 gene, is crucial for nervous system development. Its dysregulation, due to an imbalance in X chromosome dosage, can help explain the neurocognitive challenges experienced by individuals with Turner or Klinefelter syndromes. Furthermore, the ZFX gene, which also escapes inactivation, acts as a transcription factor, meaning it controls the activity of many other genes. An imbalance in ZFX can disrupt entire networks of gene expression, potentially affecting numerous developmental pathways. These examples highlight that the imperfect nature of X inactivation is not just a theoretical concept, but a tangible factor shaping the health and development of individuals with varied X chromosome numbers. The persistent activity of X-linked genes can also manifest in other specific health conditions. For example, intestinal pseudo-obstruction, a condition that impairs the coordinated muscle contractions needed to move food through the digestive tract, can be linked to genetic changes on the X chromosome, affecting the FLNA gene. The protein produced from FLNA, filamin A, is crucial for cell structure and movement, and its impaired function can affect the shape of smooth muscle cells in the gastrointestinal tract, interfering with peristalsis. Similarly, microphthalmia with linear skin defect syndrome, characterized by small or poorly developed eyes and unusual linear skin markings, is caused by a deletion in a region of the X chromosome that includes the HCCS gene. This gene produces holocytochrome C-type synthase, an enzyme vital for cellular energy production through oxidative phosphorylation and apoptosis. In females, if one X chromosome carries this deletion, the cellular mosaicism leads to an overall reduction in this critical enzyme, manifesting as symptoms. The imperfect solution of X inactivation, therefore, is not a flaw in design, but a testament to the evolutionary compromises and complexities in our biology. It creates a unique genetic landscape in females, one shaped by the nuanced expression of hundreds of X-linked genes. This landscape contributes to a distinct set of physiological strengths and vulnerabilities, influencing everything from our basic development to our long-term health. Understanding these subtle yet powerful genetic differences is crucial as we continue to unravel the intricate tapestry of human health and disease. The question then becomes, how do these complex X chromosome influences extend to other crucial aspects of health, such as metabolism and body composition? Chapter 3. The X chromosome's metabolic footprint. Consider the intricate dance of metabolism that keeps our bodies running. It involves a delicate balance of energy intake, storage, and expenditure. When this balance is disrupted, we see the rise of conditions like obesity and type 2 diabetes. What might seem like purely lifestyle-driven or autosomal genetic predispositions turns out to have a surprising connection to the X chromosome. Scientists have employed clever strategies to untangle the complex web of factors that contribute to these differences. One particularly insightful approach involves studying specialized mouse models. These models allow researchers to compare mice that have the same type of reproductive organs, meaning they produce similar levels of hormones, but differ only in their number of X chromosomes. For instance, they might compare mice with 2 X chromosomes and male gonads to mice with 1 X chromosome and male gonads. What these studies reveal is striking. Even when hormonal influences are matched, the number of X chromosomes alone can profoundly affect how much fat an individual accumulates and their susceptibility to metabolic diseases. This deeper understanding is particularly critical when we look at individuals with variations in their sex chromosome number. Individuals with Turner syndrome or Klinefelter syndrome face a significantly elevated risk of developing type 2 diabetes. In fact, studies showed they are 4 to 6 times more likely to develop this metabolic condition compared to the general population. This dramatic increase in risk points directly to the impact of having an atypical number of X chromosomes. The question then arises, what exactly is happening at the cellular level that leads to these metabolic differences? To explore this, researchers have delved into the genetic activity within various tissues that are central to metabolic regulation. They've examined gene expression patterns and other molecular markers in blood, muscle tissue, and adipose tissue, which is essentially body fat. What they've discovered is that the way genes are expressed, or turned on and turned off, is indeed altered in these critical tissues in individuals with Turner and Klinefelter syndrome. For example, studies have identified around 14 genes that show reciprocal regulation, being upregulated in Klinefelter syndrome and downregulated in Turner syndrome across various tissues. Crucially, these changes in gene expression are not uniform across all tissues. The alterations observed in blood cells might be different from those in muscle cells, and both might differ from what's happening in fat cells. This tissue-specific variation is a vital clue. It tells us that to truly understand why individuals with these conditions are more prone to type 2 diabetes, we can't just look at a single blood sample and assume it tells the whole story. Instead, a comprehensive picture requires examining multiple tissues, each contributing its unique piece to the metabolic puzzle. Moreover, the overall metabolic phenotype in these conditions is likely influenced not only by direct gene dosage imbalance, but also by additive alterations in gene networks, regulatory pathways, and individual genetic modifiers. The underlying mechanism for these metabolic effects likely ties back to the genes on the X chromosome that manage to escape the process of X-Inactivation. These escapee genes, remaining active on both X chromosomes in individuals with 2 X's, lead to a higher dose of their gene products compared to males. In individuals with extra X chromosomes, this dosage is further increased, while in those with a single X, the dosage is reduced. These subtle yet persistent differences in gene dosage, leading to a broader proteome imbalance, are believed to be at the heart of the metabolic disruptions. For instance, altered dosage of the ZFX gene can disrupt entire networks of gene expression, potentially affecting numerous metabolic pathways by organizing networks where several X chromosomal genes are highly co-expressed with several sex chromosome dosage-sensitive autosomal genes. The X chromosome is known to be particularly rich in genes that play roles in muscle function. Given that muscle tissue is a major site of glucose uptake and energy expenditure, any alterations in the function of X-linked muscle-related genes could directly impact how the body handles sugar and stores energy. If these genes are expressed at suboptimal levels or at excessively high levels, depending on the X chromosome count, it could impair insulin sensitivity, glucose metabolism, and overall energy balance, setting the stage for conditions like type 2 diabetes. This understanding opens new avenues for research, prompting us to consider sex chromosome variations not just as rare genetic conditions, but as powerful models for unraveling the broader genetic basis of metabolic health differences between biological sexes in the general population, potentially identifying novel therapeutic targets. While many of these genetic alterations logically connect to metabolic function, further research is still needed to establish the precise direct links between specific changes in gene expression and the observed metabolic characteristics in individuals with these syndromes. Chapter 4: X's Reach: Heart, Mind, and Longevity. The X chromosome plays a profound role in cardiovascular function, longevity, and the intricate architecture of our brains, leading to discernible behavioral differences. Its influence extends to these fundamental aspects of human health. The heart, a tireless pump, and the vast network of blood vessels that sustain every cell in our body are not immune to the X chromosome's influence. Recent insights reveal that the number of X chromosomes is a significant player in cardiovascular vulnerability. Having two X chromosomes can offer distinct advantages in mitigating ischemia-reperfusion injury, a damaging process that occurs when blood flow to an organ is interrupted and then restored. Individuals with Turner syndrome face a significantly elevated risk of heart defects from birth, such as specific issues like a bicuspid aortic valve and aortic dilation, which can lead to narrowing of the aorta or problems with heart valves. These are not merely cosmetic anomalies. They represent serious structural vulnerabilities that can require lifelong monitoring and intervention. Beyond congenital defects, high blood pressure is also a frequently observed issue, even in the absence of obvious aortic narrowing. Furthermore, individuals with Turner syndrome are at an increased frequency of kidney defects, celiac disease, and thyroid gland inflammation, highlighting the broad systemic impact. Similarly, Klinefelter syndrome also carries a heightened long-term risk for cardiovascular problems. Beyond these, individuals with Klinefelter syndrome face additional comorbidities such as gynecomastia, breast development, and osteoporosis, weak bones, as well as a higher long-term risk of autoimmune diseases and certain cancers. While the specific manifestations might differ from Turner syndrome, an atypical number of X chromosomes correlates with increased vulnerability in the cardiovascular system, suggesting a delicate balance of gene expression, governed by the X chromosomes, that is crucial for maintaining optimal heart health throughout life. The X chromosome's role in cardiovascular health also intertwines with its broader impact on longevity. It is a well-established observation that on average, women tend to live longer than men. While many factors contribute to this lifespan gap, differences in smoking rates, alcohol consumption, and risk-taking behaviors, there's a growing body of evidence pointing to a deeper chromosomal explanation. The findings from studies indicate that mice with two X chromosomes consistently outlived their counterparts with XY chromosomes, irrespective of whether they possessed ovaries or testes. This indicates that the X chromosomes themselves are a more significant determinant of a longer lifespan. This observation is further supported by a theory suggesting that the Y chromosome may be inherently toxic, offering an alternative or complementary explanation for the longevity difference. Beyond the physical mechanics of the heart and vessels, the X chromosome wields considerable influence over our brains, shaping cognitive abilities, emotional regulation, and social interactions, in essence, our behavior. The X chromosome is uniquely rich in genes vital for brain development and function, particularly in regions like the cortex and hypothalamus. In fact, X-linked mutations are implicated in intellectual disabilities at a rate 3.5 times higher than mutations on other chromosomes, highlighting the profound importance of this chromosome for neurological health. Variations in X chromosome number provide compelling insights into these behavioral effects. Consider XYY syndrome. Historically, this condition was sometimes associated with sensationalized and largely disproven theories of aggression or criminality. Early studies in the 1960s, observing a higher incidence of XYY individuals in certain institutional settings, led to the erroneous label of supermales prone to violence. However, extensive research has since refuted this simplistic and harmful notion. While some individuals with XYY syndrome may experience challenges, these are generally subtle and manifest differently than once believed. They may be taller than average and sometimes experience minor physical differences like hypotonia, weak muscle tone, hand trembling, or involuntary muscle movements, particularly in younger individuals. More relevant to behavior, individuals with XYY syndrome can face learning difficulties, including speech and language delays, and sometimes display attention difficulties or behavioral issues. Problems with social communication and understanding subtle social cues, such as humor, can also occur, potentially leading to social challenges. The observed higher prevalence of XYY individuals in some correctional or psychiatric facilities is now largely understood to stem from a combination of these underlying learning and social communication difficulties, which might make navigating complex social situations more challenging, rather than an inherent predisposition to aggression. The narrative has shifted from congenital criminals to a recognition of specific neurodevelopmental profiles that require understanding and support. Similarly, females with triple X syndrome often present with a distinct set of behavioral and cognitive profiles. While many individuals exhibit no noticeable symptoms, others may experience developmental delays, particularly in speech and language acquisition and fine and gross motor skills. Specific physical features can include epicanthal folds, widely spaced eyes, curved pinky fingers, flat feet, an inward bowed breastbone, and poor muscle tone. Learning disabilities affecting academic areas such as reading, understanding mathematics, or written expression are also more common. Beyond academic challenges, behavioral and psychological differences can emerge, including a higher incidence of attention deficit hyperactivity disorder, symptoms suggestive of autism spectrum disorder, and increased susceptibility to anxiety and depression. These girls and women may also experience difficulties with memory, judgment, and information processing. The stress of grappling with these differences can also contribute to issues with self-esteem. The dosage effects of sex chromosomes also manifest in cognitive profiles. Studies examining individuals with even rarer forms of sex chromosome aneuploidy reveal a clear correlation. Each additional X or Y chromosome is associated with a measurable decrease of around 1 standard deviation in IQ. Intriguingly, the nature of these cognitive impacts can differ slightly depending on which sex chromosome is extra. Additional X chromosomes tend to have a greater effect on structural language skills, influencing grammar and sentence construction. In contrast, additional Y chromosomes are more disproportionately linked to pragmatic problems, which involve the social use of language, and to features often associated with autism. Interestingly, XXY and XYY boys often exhibit similar communication profiles, whereas XXX girls tend to show less evidence of pragmatic impairments. This nuanced understanding highlights that the specific genetic content of each sex chromosome contributes distinctly to the intricate wiring of the brain. Even in Klinefelter syndrome, neurodevelopmental differences are common.
Individuals may experience learning difficulties, particularly in language and academic achievement, and some behavioral concerns. Psychological challenges such as depression, anxiety, low self-esteem, and mood instability have also been noted. While the intellectual impact is generally mild unless more than 2 X chromosomes are present, the subtle alterations in brain function can affect social development and emotional well-being.
The X chromosome's profound role in brain function is also demonstrated by the sheer number of genes on it that are expressed in brain tissues. Research indicates that the X chromosome accounts for approximately twice as much brain variation in males compared to females, and 4 times more genetic associations related to brain traits are identified in males. This suggests a complex interplay where the single X chromosome in males might have a more direct and less buffered impact on brain development. Furthermore, a specific region on the X chromosome, known as XQ28, has been found to be particularly rich in association signals for brain imaging traits, and is linked to various intellectual and developmental disabilities. This region contains 5 specific genes, AM3A, PJA1, TMLH, PLXNA3, and ZNF275 that impact brain structures and functions, schizophrenia, autism, and educational attainment. These examples, along with the numerous other X-linked genes involved in neural processes, collectively underline the X chromosome's critical contribution to our cognitive landscape and behavior. The subtle hand of the X chromosome, therefore, reaches into the very core of our physiological and psychological being. From the rhythmic beating of our hearts to the complex tapestry of our thoughts and emotions, its unique genetic endowment shapes vulnerabilities and differences that are only now beginning to be fully appreciated. The challenge ahead lies in unraveling the precise molecular mechanisms by which these X-linked genes exert their influence, promising to unlock new understandings of sex-specific health and disease and pave the way for targeted interventions.
Chapter 5. Mosaicism's Double-Edged Sword. The intricate process of X-Inactivation, a biological marvel we've explored in earlier discussions, doesn't just switch off one of the 2 X chromosomes in females. Its truly random nature in most cells creates a fundamental difference between individuals with 2 X chromosomes and those with only 1. This difference manifests as a profound cellular diversity, a phenomenon known as mosaicism. The body of a female is a cellular patchwork woven from countless cells, each carrying either the maternal or paternal X chromosome as its active blueprint. This unique characteristic, absent in individuals with a single X chromosome, profoundly shapes female health and disease in ways that can be both remarkably advantageous and unexpectedly challenging.
This mosaicism acts as a powerful buffer, a protective shield against many X-linked recessive conditions. Consider a scenario where one of the X chromosomes carries a faulty gene, perhaps one responsible for a critical function. If this gene were on an autosome and the individual inherited 2 faulty copies, the consequences could be severe. However, in a female, even if one X chromosome carries a pathogenic variant, the random nature of X-inactivation means that approximately half of her cells will have the healthy X chromosome active, producing the necessary functional protein. The other half will have the X chromosome with the variant active, but the contributions from the healthy cells can often compensate, preventing or significantly reducing the severity of the disease. This is why females are frequently carriers of X-linked recessive conditions like hemophilia, color blindness, Duchenne muscular dystrophy, DMD, or Becker muscular dystrophy, BMD, rather than being affected themselves. They possess a built-in biological workaround, a cellular redundancy that males with their single X chromosome simply do not have. If a male inherits a bad allele on his X chromosome, he has no second copy to fall back on, making him much more susceptible to expressing the condition. This protective aspect of mosaicism highlights a remarkable evolutionary advantage, transforming a potential genetic vulnerability into a source of resilience.
Beyond individual gene defects, this cellular diversity also contributes to the female immune system's renowned robustness. The X chromosome is rich in genes that play vital roles in immune function, and the number of identified X-linked genes and microRNAs involved in immunity continues to grow. With 2 X chromosomes and the mosaic expression of different X-linked genes across various immune cells, females essentially have a broader repertoire of immune responses. It's like having a diverse team of defenders, each trained in a slightly different style of combat. This enhanced immune activity is believed to contribute to females' generally better survival rates against infections compared to males. The mosaic expression allows for a wider range of physiological responses to various stimuli and injuries, providing a flexible and powerful defense mechanism against external threats. Even the supposedly inactive X chromosome, while largely silenced, isn't entirely discarded. It remains within the cell and can, in some instances, act as a template for repair if the active X chromosome sustains critical damage, adding another layer of cellular backup. However, this cellular diversity, while offering significant benefits, also presents a complex set of vulnerabilities, acting as the other edge of Mosaicism's sword. The beautiful randomness of X-Inactivation is not always perfectly balanced. Sometimes, by chance or other mechanisms, one X chromosome is preferentially inactivated over the other in a process known as skewed X-Inactivation. If the X chromosome carrying the healthy version of a gene is disproportionately silenced, then the X chromosome with the pathogenic variant becomes active in a larger proportion of cells. In such cases, a female carrier, who would normally be protected, might begin to manifest symptoms of the X-linked condition, sometimes mildly, sometimes more severely, depending on the degree of skewing and the specific gene involved. This means that the protection offered by Mosaicism is not absolute and can be undermined by this deviation from random inactivation, primarily impacting the manifestation of X-linked recessive conditions.
This delicate balance is particularly evident in the realm of autoimmune diseases. While the robust female immune system offers protection against infections, it also comes with a hidden cost, an increased susceptibility to conditions where the immune system mistakenly attacks the body's own tissues. The very strength and diversity that make it effective against pathogens can, when misdirected, lead to self-destruction. Skewed X-Inactivation, or even the reactivation of specific genes on the otherwise inactive X chromosome, is thought to play a role in this predisposition. If certain immune-related genes, expressed in a mosaic fashion, lead to an immune response that is too aggressive or misidentifies self-components, it can contribute to the breakdown of self-tolerance, paving the way for autoimmune disorders. The X chromosome, therefore, is intimately involved in this double-edged aspect of female immunity, contributing to both superior defense and heightened autoimmune risk. The variable expression introduced by Mosaicism can also lead to a wide spectrum of symptoms in certain X-linked conditions. In females, because of X-Inactivation, some cells will produce a necessary protein from the healthy X chromosome, while others will produce none from an affected X. This cellular mix-and-match results in an overall reduction of the protein, leading to the manifestation of a syndrome. The precise pattern and severity of symptoms can vary greatly depending on how this mosaicism plays out in different tissues and individuals. Mosaicism also adds a layer of complexity to conditions involving an atypical number of sex chromosomes. While previous chapters discussed the impact of having an extra or missing X chromosome, these conditions can sometimes occur in a mosaic form, where only some cells in the body carry the chromosomal abnormality. For instance, in mosaic XXX syndrome, only a portion of a female cells will have 3 X chromosomes, while others will have the typical 2. Similarly, mosaic Klinefelter syndrome means that only some cells in a male will have the XXY configuration.
In these mosaic cases, the symptoms are often milder than when the aneuploidy is present in all cells, because the presence of normal cells can partially compensate for the abnormal ones. For example, some individuals with mosaic Klinefelter syndrome may have less severe signs and symptoms than those with the extra X chromosome in all their cells, depending on the proportion of cells with the additional chromosome. The same applies to mosaic Turner syndrome, where some cells lack an X chromosome while others are normal, often leading to a less severe presentation than complete Turner syndrome. This variability in cellular composition directly influences the severity and specific presentation of these complex genetic conditions.
A particularly intriguing and concerning aspect of mosaicism emerges with aging. As women grow older, a phenomenon known as mosaic loss of chromosome X, or MLOX, can occur in their white blood cells. This means that a significant percentage of these immune cells may spontaneously lose one of their X chromosomes, leaving them with only one. This isn't a completely random event. Researchers have identified 56 common genetic variants and rare variants in a gene known as FBXO10 that can influence this loss. Crucially, these variants are often found on the retained X chromosome, suggesting a growth advantage for those cells that have lost the other X. This proliferation of white blood cells lacking an X chromosome raises the risk of developing certain health conditions, including blood cancers. It's a striking example of how a process that initially fosters cellular diversity and resilience can, under specific circumstances, become a pathway to disease, illustrating the delicate balance inherent in female biology.
Furthermore, recent research is revealing that the inactive X chromosome is not merely a silent, passive partner. Instead, it appears to actively modulate the expression of genes on the active X chromosome. This means that some genes on the inactive can influence whether their counterparts on the active X are dialed up or down, adding an unexpected layer of regulatory complexity. In fact, studies suggest that 38% of the X chromosome genes are affected by the presence of inactive X chromosomes, either because the genes are expressed on the inactive, or because the inactive regulates their expression on the active X, or through some combination of the two mechanisms. This intricate interplay between the two X chromosomes, far from a simple on-off switch, contributes to the unique physiological characteristics observed in females, and further complicates our understanding of how X-linked genes influence health and disease. This dynamic interaction means that the cellular diversity created by mosaicism is not static, but constantly evolving, with profound and still largely unknown implications for the lifelong health trajectory of individuals with two X chromosomes. How many more layers of complexity will we uncover in this remarkable dance between the active and inactive? And what will that mean for personalized medicine and our understanding of sex-specific health? So, we've journeyed through the intricate world of the X chromosome, revealing that having two X chromosomes is a biological tightrope walk. While X-Inactivation largely balances gene dosage, the persistent activity of escapee genes and the dynamic nature of mosaicism create a unique genetic landscape. This intricate interplay shapes female health, influencing metabolism, cardiovascular function, brain development, and even longevity, often providing resilience but also specific vulnerabilities. Understanding these nuanced genetic differences is crucial for unraveling sex-specific health and paving the way for personalized medicine. Thank you for listening as we continue to unlock the profound complexities woven into our very chromosomes.